Let me welcome all of you to the seventh day of this Institute Seminar Days 2020-2021. From the organizer's side, I would like to thank everyone who has participated, especially those who gave talks and have shown interest in this event. I welcome you all to today's Seminar Days. It's an exciting day because we have two talks lined up by Professor Vani Vemparala and Professor Rahul Sinha. Vani would be talking about designing biomimetic polymers, antimicrobial agents. Vani, looking forward to your talk. Thank you for doing this.
Thank you. Thanks, Arijit and others for organizing this. So, I thought before I start talking about the particular problems of interest to me, first thing I want to say is that I tried really my best to make it as non-technical as possible. I think there's like one equation in the entire slide. But I also thought that before I speak about the problem itself, I should motivate by saying what are the length and time scales of the problem that particularly interest, especially given that we've had some talks before. I just wanted to place myself, where is it that my research lies? So here is a very generic slide about, different length scales and different problems of interest. We've had Manjari talk back in November, and we had Pinaki had spoken, Apurva, they more mesoscale kind of things. And then we had Indumati, and we're going to have Rahul after my talk, and Aparna. These are the talks I've seen from our IST website, and these are the different time scales where I think roughly all the problems that they do lie in. And I just wanted to motivate as to where exactly the kind of work I do lies. This is typically the length scales of the problems. So the length scales I'm interested are probably ranging from nanometer to micrometer, and that is where most of the problems that I'm interested lie. Okay. So having motivated this, I just wanted to add further as to exactly what kind of problems can be done today, not just by me. This is a very generic slide. In general, if one is interested in doing atomistic simulations of soft matter or biological systems, what are the length scales? This is very important to understand, to put in perspective what kind of calibrations or what kind of things that one can do. So here you can see, mechanical pencil 0.5 mm, right? Compared to that, if you take an entire cell, the size of the cell is typically 10 to 100 micrometers, right? But now if I take a small portion of that cell and then zoom it in, which is like your cell membrane. Cell membrane is something that sort of envelopes contents of the cell. And there the thickness we are talking about 5 to 20 nanometers. This is nanometer I've given, it's about 10 power minus 9 meters, right? And if I further take another small portion of this and zoom it into that, you get something called a phospholipid bilayer, which looks somewhat like this. Basically, there are some molecules called lipid molecules which self-aggregate, and they form these kind of a bilayer structure which envelops the contents in the cell, right? And if I further zoom into one of those things and then look at in detail how that molecule looks like, this is how it looks like, and the length scales we are talking about is 10 to 20 angstroms. And from your high school, you may remember that a typical carbon-carbon bond length is about 1.5 angstrom. So these are the length scales that we are talking about, where most of the things that I'm interested happen, right? So before going, I just wanted to give you a very brief thing about something that, some kind of a jargon that we will keep talking about. If you remember, I showed you a small lipid molecule. You can imagine this as consisting of two things, a part that loves water and a part that hates water, right? This is the blue is shown as a thing that loves water, and the other part is the one that hates water. Now, you may think this is a very simple description, but this is entirely responsible, for example, how cells are made, how you and I are made, and everything else. Because, as you can imagine, if you take water-loving things and add it to the water, they dissolve in it. Basically, they distribute themselves. If you take things that don't like water, they tend to come together, like you can imagine as oil droplets, right? This very, very simple behavior of molecules as their response to water gives rise to very interesting things. So suppose if you take a bunch of these things and drop them in water, they can form all kinds of structures. So one of the structures they form is something like what is shown on the right hand side, right? So what they're basically trying to do, all the molecules, the water hating molecules are trying to hide away from water, and they are inside, and all the water loving molecules are outside. Now you can imagine water as on the top and bottom of this. But still there is a problem because the things that are sitting on the side are exposed. So the way they minimize this exposure to water is they try to bend and they form these seal compartments. And that is what you realize or we recognize as cell. So this very fundamental aspect of things loving water, not loving water, is something very fundamental to biology, which determines a lot of things. For example, you might have heard of things like protein folding. At least a large part of the protein folding is driven by something called this hydrophobic and hydrophilic interaction. So this is just something I thought I'll just put it out there too, because we'll be referencing to this language before later. Okay, then let's come back to a second part of the problem, which is that there are different kinds of cell. Again, this is, I'm not a biologist, and I'm saying this from a very coarse grain point of view. So what kind of cells we are interested in is typically you have a bacterial cells. These are most of the microbes that, let's say, pathogens. And then you have a host cell, which is, let's say, you and me, animal. So what are the differences between these two cells which are of relevance to us or relevant to me as a physicist in probing certain things? So the things that are relevant is that if you take a host cell, like any animal cell, it's an overall neutral, which is if I'm looking outside and looking at this cell, it looks neutral to me. But if I look at a bacterial cell, it has certain kind of negative things that are sprinkled on it. Now, again, I'm saying it in a very technical, non-technical term, but just remember that this is a fundamental difference between a neutral, a host cell, which is an animal or a plant cell or anything, and a bacterial cell. So now we come to the problem motivating as to why we are interested in this class of things called antimicrobial agents. So as you all may know, and you might have even used them, there is a huge problem with the usage of antibiotics today. You might have heard of things called, resistance, bacterial resistance, and things like that. So before we even go to that, if we just try to understand how antibiotics actually work. As I said before, there are some fundamental differences between a bacterial cell and a host cell, and these antibiotics preferentially, they try to target this by exploiting the differences between the two. Not only do they target these things, what they eventually end up doing is that they actually target very specific process, which means, for example, something like a synthesis of a cell wall, or let's say a protein or a DNA replication. These are very, very specific processes that these antibiotics actually target. But if you see this graph, this is a very nice graph, which shows on the top the timeline and the top of all the kinds of different antibiotics that have been introduced to in clinical usage, and the bottom of it shows when the first resistance of bacteria was observed. For example, if you see something like erythromycin, which is usually what we take for something like sore throat or something, this was introduced back in mid-1950s, and within 30 years, you could actually see that there is a resistance for bacteria. What is resistance? What we mean by resistance typically in this setting is that you need stronger and stronger antibiotics to kill the same kind of bacteria, right? Now, this is clearly a problem because these antibiotics are putting extensive selective pressures on these microorganisms. Basically, only those organisms which survive these antibiotics will keep procreating, and then you have to use stronger ones to kill them. And then there is this there is actually a great risk of having an outbreak where you cannot even have any antibiotic which can have a lot of effect on these things. So what has been for last 20 years or so, 20, 30 years, there's been a lot of push in trying to understand, can we design, now maybe antibiotics or making stronger antibiotics is not the way to go about. There are clearly a lot of issues. So can we then go back and design, or we should have a paradigm shift in looking for new kind of microbial agents, right? Hopefully these are the ones which have lesser bacterial resistance issues, or they significantly delay, in the sense that, let's say, instead of 30 years, you have bacterial resistance over maybe let's say for 100 or 150 years, you're still delaying and the clinical usage will still be good, right? So that's the aim and that's the motivation for what we are trying to do. So if I just go back to the basics, your immunity, whatever is it, can be divided into something called innate and adaptive. Innate is something which is very immediate. It is a very like a frontline soldiers. It's not very sophisticated. It only says this is the animal cell and I recognize this is a foreign cell and I'll kill it, right? There's an extremely coarse-grained thing. Adaptive is something which is much more intelligent. It looks at what pathogen has come, devises a specific methodology to attack it and things like that, right? What we are interested is in this innate immunity. So what people have recognized, started looking at in 1980s is that a lot of amphibians, basically a lot of frogs, on their skins, they have a lot of these proteins which basically kill the pathogens and allow them to survive in probably the dirtiest and the filthiest waters around, right? And so people started asking this question as to what are these special antimicrobial peptides which allow them to do this? How are they doing? Can we understand something from these naturally occurring ones? And can we use them in a drug, in a clinical setting, right? That is the goal. The goal is because we already have this. For example, even in humans, you typically tend to have this in the gut lining or in your tears or in your saliva and things like that. So these are already present. These are a part of our own immunity system. But the goal is now, can we understand how these are designed? How do they function? So that we can maybe put them in some sort of a clinical use, right? So that's a very different way of doing. Then one may ask a question, why are they effective? Why are they more effective than antibiotics, right? Because that's a goal we are trying to go. We are trying to have a paradigm shift in learning. They are more effective for a very simple reason among many. It is that they don't act on any specific target like antibiotics act. They are very generic sort of killers. So they target the cell membrane. Once a cell membrane is targeted, they can go and target DNA. They act on multiple targets. Now, the problem is that if bacteria wants to develop a resistance against these agents which act on multiple things, then either the mutations that they need to undergo have to be very extensive. They have to probably reorganize their cell membranes completely. Any of these processes that we are talking about in times of length scales takes much, much longer than any resistance that has been observed for antibiotics. So somehow these agents, which nature has of course developed over millions of years, have optimized such that bacteria is somewhat of ineffective in front of these things. Of course, there are resistance to these things, but I wouldn't have time to go into that. But these seem a very promising agents in which we can, because they act so coarsely, they are there's no sophistication. They basically I'm going to show how they basically just tear apart the bacterial membrane, right? So they're very coarse agents. And so the goal is, how do we understand these things and how do they work? So remember, I told you that your mammalian or your host cells is overall neutral, and your bacterial cells have something called this negatively charged lipids that are sitting on top of it. So the nature is a very wonderful thing, and what they have developed is that whatever these antimicrobial agents which are found in nature, they have evolved such that they have a lot of positive charges on it. So you can see very electrostatically, if suppose you have a human cell and you have a bacterial cell, these things can differentiate between these two simply because one is negative and the other is neutral, right? So it's a very, very simple way of recognition between the two. Once it recognizes and then latches onto that, so you can see that, for example, this one, where this is a bacterial membrane, and this interacts with this very strongly in terms of electrostatic interactions. And then in terms of, let's say, human or a mammalian membrane, the interaction is only what is called a hydrophobic, which is just, you can think of it as a, not really van der Waals sort of interaction, but a much weaker interaction than electrostatics. So how do they work and they kill this bacterial membrane? As I said, they are very beautifully designed so that they detect the bacterial membrane because they have oppositely charged. Once they detect, they sort of aggregate onto that membrane, and they can tear the membrane apart in many ways. For example, they can form pores, and you can imagine like a balloon, and you have a pore, and then balloon just deflates and everything inside the bacterial cell oozes out and the bacteria dies, right? So as I said, extremely coarse-grained way of killing bacteria, right? So then one may ask a question, so these things are available already. You have all these wonderful killers that, available in you and me and seem to be doing well. Then why are we interested in developing polymers, right? The reason why we are interested in developing polymers is that if you want to have a clinical implementation of these peptides, as I understand from my experimentalist collaborators and others, there is a huge issue that they are susceptible to, okay, a term this proteolytic degradation is nothing but some kind of bacterial proteins which go and tear this peptide apart. They basically break it down. So you have all these kind of issues, and there are issues also of something called bioavailability. Bioavailability is you take X amount of some drug or a medicine, how much is eventually available for your body. So there are a lot of issues with these naturally occurring ones, and probably producing them on a mass scale is probably much more expensive, and it's also not something that there is a right handle to do exactly what is it needed. But on the other hand, polymer industry is very well developed. People know exactly how to make these polymers, and then the idea is to understand at a very low cost, can I develop a synthetic polymer which mimics these naturally occurring peptides, right? So that's the question that one has in mind. But here there's something I want to give a caveat. You can already imagine most likely that this is going to be a very difficult task. Nature had millions of years to develop these things. We are trying to develop polymers in a lab on an extremely short time scale compared to nature. As I also understand from my collaborators, it's very easy to design a polymer that kills everything, that kills human cells, bacterial, everything. But to introduce that nuance into this polymer so that it recognizes the difference and then attacks only the bacterial is something which is a very difficult task. And the experiments are also not very precise that they can understand, and that's where simulations come in, because simulations we can go at an atomistic scale and try to understand what is it that they're exactly doing, right? So the goal is generally is this antimicrobial peptide. This is the naturally available one. I want to design an antimicrobial polymer which is a mimetic design, which is it behaves like that antimicrobial peptide, right? So here is a very fancy thing. We had this as a cover article in one of our things. So the goal, what we are trying to do is that you have this bacteria and we want to design all these different polymers which go and attack and basically kill this bacterial cell, right? So that's the goal. And also to ask a question, how do you use very simple design of polymers that give certain structures that are available? What I'm showing is two examples of antimicrobial peptides, and what is the difference between them? One is a very linear kind of a thing, one is a very, what do you call, a collapsed globular kind of a thing. How do you design polymers which go from one to the other, right? So the goal basically is to design polymers that capture essentially the physiochemical properties, but we are not interested in exact chemical structure. It's probably not possible. But what we're trying to do is, can you get roughly what this antimicrobial natural peptide is doing? Can I design a polymer to do that, right? And the aims, there are many aims. One is to understand how do these things work? How do they attack a bacterial membrane? What are the kind of tweaks I have to do so that they are more effective against bacterial membrane? But if I use it against, let's say, mammalian membrane, nothing happens. That's what you want, right? If you can give a large quantity of these drugs to, let's say, humans, nothing should happen. But even if you want to give the smallest quantity, and they should attack the bacteria. So this is the kind of very nuanced thing, what we need to get, which is high selectivity against bacteria and very low toxicity to humans. Right? That's the goal. So now, as I said, this is how a typical peptide looks like. So if I want to do a very bare bone design of it, I want to take a polymer and I will want to put exactly in this manner, right? So that's because that's exactly looks like that. But this is extremely expensive to design. Please remember that anything in which you want a specificity is difficult to design. Is very expensive. Everything is difficult because you have to be very, very specific about what you put in so that you get that shape. Extremely difficult. But the other kind of polymers where, they are jumbled up randomly, very easy to prepare, right? Then the question becomes, I know that if my polymer has to behave like this peptide, it should get this shape. But I cannot put that shape in hand because it's a bit expensive. People have tried. There are also polymers which work on this. But I'm more interested in asking the question, can I make a random polymer which is easy for me to make, but somehow make it smart enough that when it goes and attacks a bacterial membrane, it takes the shape of this, right? So that is the goal, which is I take this peptide, I design a polymer, then it goes and attacks the membrane, but it eventually takes this shape. So basically, I want to design a polymer that is random in shape, but when it is interacting with the bacterial, I want it to acquire this shape. So how do you put this smartness into this polymer that it recognizes this thing, right? So that's the questions that I'm trying to ask. So this is basically the different kind of very chemical structures of the polymers that I have been working on for quite some time. I have an experimental collaborator who does these experiments in the lab, and we try to do the simulations and we try to validate back and forth and try to come up with basic understanding of how do these things work. So very, very single slide. This is the only slide which has an equation. So the simulation methodology is I do something called atomistic molecular dynamics simulation. So basically we ask a question, we build a model, we put a lot of interactions to exactly mimic the systems, and then we solve the equations of motions. In this case, what I do is called classical simulations. So I do Newton's equations of motion. Everybody +2 level knows this. We just solve equations of motion, but of course the most time-consuming part is in this interactions I have to put in, because I have to tell the computer that this is a polymer, this is a lipid membrane, this is a bacterial membrane, this is water. That is where all the complication of the things lies. But the solving of the equations is very simple, right? So let's assume we do that. Now, I just want to answer a question as to, again, a question why people want to do simulations of these things. So the experiments are very crude. What they tend to do is they take something like an E. coli bacteria, they put some probe inside, a fluorescence probe, which means it lights up when it comes out. So then they inject some polymer solution, and if this polymer solution kills the bacteria, the fluorescence probe comes out, they can record and they can say that the bacteria is dead now. So you can already see these experiments are very coarse. They really do not know what is actually happening when these polymers are interacting with the bacterial membrane. They only know that they kill these things, right? So this is where simulations can really help, because we put all gory details of every at atomistic level, and we can give back some information to the experimentalists which are very difficult to probe. So as I said, the questions of interest for me as a physicist is, how do these polymers interact with these membranes? What happens to these structures when they go from water to membrane phase? What happens to the membrane itself when these things interact? And how do they differentiate basically between a bacterial membrane and a cellular membrane? Is it just the charge? Is it something else? These are the kind of questions that I'm interested in. So this is the typical way we set up a simulation. We take a polymer, a single polymer. This is exactly what experimentalists do. We simulate it and form an aggregate of it, and then we put in a bacterial membrane mimic, and we put water, and then we do full simulation. So what I thought I'll later show is just a couple of movies so that I can sort of make this a bit more interesting. And you can already see that when we put bunch of these polymers, depending on what we put, they take different shapes. These shapes are very important because depending on the shape, the way they interact with the membranes are very different. So we can look at all these kind of aspects where we keep changing the polymers and look at how they do. So maybe I'll show a few movies and then probably end this by just saying what are the kind of things that we know today. So here what I'm trying to show you is that this is a polymer which has only two kinds of groups. The red one is a what is called a hydrophobic group, and the green one is what is called a charged group. So if I now run the movie, so I probably, let me just go back to the beginning, and I want to just get your attention. Sorry. I want to get your attention before starting that, as you can see, this is completely random in its shape, right? There's red, green distributed all over. Now, please remember the shape, and we'll see how this evolves as it is. What is shown below is a snapshot or a zoomed in version of a bacterial membrane. Once it goes inside the bacterial membrane, please see how the shape actually changes. So I can make this faster. And if you see here, this shape is completely different from when it started. Okay, so if I want to show, this used to be a very randomly distributed, but once you come to a membrane, it takes the shape which is distributed along this. So all the greens are pointing in one direction and all the reds are pointing in one direction. And if you remember from a couple of slides back, this is one of the shape of the antimicrobial peptide that is naturally available, where this division of positive and this. So with a simple molecule with two shapes, we can actually imitate a peptide that is naturally occurring in nature. How is it that it's going to take the shape? So basically, I'm not showing it here, but if I pull this polymer out of it, it again falls back into random copolymer, which means somehow there is an intelligence built into this polymer, which recognizes that if it is in a bacterial membrane, it should take a functional form which allows it to do a function. But once it comes out of the bacterial membrane, there is no need. So there is no need to build in a structure a priori, but these are smart enough to detect the environment and take the particular forms, right? But now if I add one more group to this, right? Here again, it is more or less random, but if I now add a third group to that polymer and do the design and do the simulations, these take completely different shapes, right? For example, this is an initial thing on the left, and on the right, if you see the final one, it is almost taking a collapsed and almost a circular kind of a shape. So by just changing and adding a new kind of a functional group, the form that these things take completely change, right? So if you remember, I just want to go back to this, where we have said, can we design simple polymers to mimic this kind of a thing? You have one which is very, elongated and all the distributed on both sides, and one which is a collapsed one. And as you have seen, with using two components of the polymers, we are able to mimic this naturally occurring peptide, which behaves exactly like the structurally, which is like this. But now I can fine-tunely add its third component into this polymer so that it can now behave like another class of antimicrobial peptides, some family called defensin. Right? So we now seem to have a sort of a control over what kind of things we want to put into this polymer, so that eventually we get the kind of shapes that we want, so that these are more effective. So the way these work is, our experiments tell us, experimentalists, collaborators, they tell us these are the kind of polymers they are interested. We make small tweaks. We run these simulations. We tell them back that maybe if you make these changes versus those changes, if you add this kind of functional group to the other, how are the things that are going to change? And eventually this is kind of a circular thing where the information goes back and forth, and a more fundamental understanding of how these polymers work is going to be hopefully evolved. So I just want to thank you, all of you, for listening to this. I tried my very best not to go into any technical details, because I think I'm available in IMSC. If someone is interested, papers are available, they can read. But I hope I just conveyed a story of how this design goes into this, the polymer design goes into designing a lot of these peptides. And this is quite an exciting field, because we're just scratching at the beginning. There's so many things that we still don't know. We are now doing some other work with our postdoc Sushmita Ghosh, where we are trying to see how these polymers interact with DNA. So that's again another very interesting thing that happens. But there are a lot of exciting stuff that can happen when you go about designing polymers, keeping biologically available ones in mind. So I'll end my talk here, and if you have any questions, I'll be very happy to answer.
Thank you, Vani, for this wonderful talk. Yeah, so Rahul has already raised his hand. So Rahul, please go ahead and ask the question.
There's not really a physics question, but if you have a very powerful broad spectrum antimicrobial drug, it will also damage the innate microbiome in your body, right? So, which is also important. So, which is also a problem with powerful antibiotics. So, any comments about that?
No, so again, as I mentioned in the beginning, so what I understand is that the goal, that is why, is not to have stronger and stronger antibiotics, because I think not just the resistance, of course, the problem you mentioned, but I think that's why there is some interest in fact, as I understand right now, on this antimicrobial peptide kind of a front, there is one drug that is about to come, which is more like a foot cream. It's not still yet into the invasive. You don't actually take it in. But I think these are much milder, and I think the resistance that they develop is much lower. So the hope is that the kind of issues you refer to hopefully do not arise in these kind of settings, or at least, let me put it this way, they get delayed in these settings. So they also, by the way, undergo resistance. This bacteria develops resistance against these as well. It's just on a different time scale. So I think that's what people are trying to exploit. Okay. So I agree with you that it's an issue. That's what I'm trying to say. So hopefully these may not be those kinds of things.
Okay, thanks.
So next Ronnie, and after Ronnie, Sayantan, and after Sayantan, Amir. Ronnie, please.
Yeah. Hello. Am I audible?
Hi. Yes.
Yeah. Thank you for the wonderful talk. So my question is that when you are saying that this is the kind of experiments that can be done presently are crude, I mean, what does a good experiment suggest in this case? We already know that these AMPs, they bind membranes and they disrupt them. So what else do we want to see from a good experiment?
So what we don't exactly know is that, for example, so in experiments, what they try to do is they develop these polymers. Let's say I've not gone through that, but you can have, let's say, I can ask a simple question. Should I design a polymer which is 8 monomers in length or 16? Is 8 better effective than 16? So these are the kind, or why is it effective? So, so the question of why is something that is never exactly answered in the kind of experiments, because they only know that one is more effective than the other. But if I don't understand, let's say if I change, of course, I have to use a technical thing. If I use methyl versus, let's say, some other kind of a functional group, what actually happens is something that's not very clear, because there is also need to understand what is actually happening when these things do. Yeah. But that you really need large, probably very refined experiments to do sort of single molecule sort of experiments to probe these, which are very difficult to do.
Yeah, so, very likely lots of cryo-EM to take free snapshots of.
Perhaps, right. So, these are the issues that experimentalists face. So they know something is happening, but they don't exactly know what actually is happening.
Another question I have is, when you are running these simulations, do we take into consideration if we have like multiple molecules of these polymers, do they aggregate?
Yes, of course. I can't show all this. Yes, of course. The answer is yes. So in fact, it's very clear that a single polymer acts on these membranes in an extremely different way compared to if you have multiple ones. Very different. Yeah.
Very interesting. Yeah. Thank you.
Yeah.
Sayantan, please.
Thanks, Vani, for this nice talk. So I was wondering, is there any uncertainty in the potential that you choose in the simulation walls, or it depends on the type of problem you're interested in?
So the answer is not all the times the potential that one is interested is available. What I mean available is that a lot of these are generic enough that sometimes somebody has developed the potential for this. But a lot of times if you are looking for a new polymer molecule, no potential is available. In such cases, there is no other option but to do some kind of a quantum calculations, like a density functional theory kind of calculation, small ones on a single molecules, and develop your own potential. And then, of course, you try to validate it against some experimental data or some kind of a physical or a biological intuition that you have. And then you try to optimize that. But yes, not all the times potentials are available. Sometimes you have to develop on your own. The one additional thing I want to also say is that sometimes maybe the very specific ones may not be important. So in this atomistic simulation, all specific details are important, but sometimes people do what are called coarse-grained simulations, where all the details are, sort of blurred, are renormalized. In such cases, maybe the detailed ones are not relevant. So it depends on what kind of question one is interested in asking. But there are tools to develop if one wants.
It may be very complicated also, right?
It's very difficult to develop, because once you develop something for one polymer, if you want what is called a transferable thing. If that same polymer exists in something else, you want to be able to use the same thing. Otherwise, you have to develop all of these potentials again. Extremely time-consuming thing. Yes.
Amir?
Thanks.
Yes, ma'am. Actually, you showed in the very first movie, like you started with a random polymer, and when it interact with the membrane, it's take a specific shape. Like all the charges are one-sided. So I was
Yes.
asking, like, what is responsible for this behavior? Like you said, there is some intelligence, so we are looking. So what are the
Yeah.
factors which lead to the this? So what is exactly responsible? Is it a charge, electrostatic interact, like?
So it's different things. Again, I didn't have time to go through much. So the bacterial membrane, actually, it has some negatively charged lipids, which I mentioned before. So what they try to do in the simulations, we very clearly see in multiple papers, we have seen this, is that the polymer first sort of latches onto it using a charge. Once they do that, what they seem to be doing is that they are making the different negatively charged lipids move into one location. So there's some sort of a phase separation that is happening. And once that phase separation happens, there are these domain walls that form between two different domains. And these polymers are smart enough that they recognize there is a weakness, and then they try to insert themselves into that. And once they insert, the charged ones want to interact with the charged head groups of the lipid, and the interior of the membrane, I mentioned in my few slides that it is hydrophobic. So the hydrophobic part wants to interact with the hydrophobic. It's like snorkeling, you have these charged ones holding on to the top and the feet sort of inside the water. So they take that shape because that is their most optimally free energy minimized shape that they can take. But before they do that, they really do completely change the cell membrane into different domains and introduce phase domain boundaries and a lot of weakness. So basically, these are the free energy minimized structures if you have an environment which has charge on the top and hydrophobic in the bottom, as is seen in the membrane.
Okay. And also, so this like, does it have a memory? Like, will it be effective for different types of membranes? I don't know whether we have different type of. So I'm just asking.
So we are doing some other work now with something else, which is these HIV peptides, how they interact with host cells. And depending on, by the way, bacterial cells don't have any cholesterol in it. So depending on the amount of cholesterol, these peptides can interact very differently with different ones. So you are right, it happens. But bacterial cells are more or less the configuration is fixed. There's no cholesterol or anything like that. So these shapes probably will be preserved in these bacterial cells.
Thank you.
Thank you for this wonderful
Thanks.
talk.